Blood. Blood is a connective tissue that carries and transports oxygen, carbon dioxide, minerals, and energy to all parts of the body. Hematology is the study of blood and its disorders. So, in today's video, I will specifically talk about blood. Composition of blood. Blood carries 45% red blood cells, 55% plasma, and about 1% white blood cells and platelets. Red blood cells are also called erythrocytes, whose main function is to carry oxygen to all parts of the body. Then white blood cells are also called leukocytes, where its main function is to fight against infections, while platelets are also called thrombocytes, whose main function is to form a clot at the site of a damaged vessel. Blood also contains antigens and antibodies which help fight against foreign particles. The antibodies bind themselves to the surface of the antigen, acting as receptors, helping in recognizing the type of infection. Our blood contains millions to trillions of cells that cannot be seen by the naked eye. So to see it, specific type of tests are done to figure out specifically what is the condition of your body, from common disorders like anemia to diagnosing severe cases like blood cancers, including leukemia and thalassemia. So the first thing to do in order to observe blood is to get some. And to do that, we perform something called venipuncture. Venipuncture is the procedure widely used in the field of phlebotomy to draw blood for diagnostic purposes. The blood is commonly drawn out from a vein through your body to store it temporarily in order to perform several tests to determine your diagnosis. So, to show how phlebotomy is done, I got out an IV injection pad to resemble phlebotomy. Here, these resemble veins where to draw blood, first you have to feel the vein by physically feeling the flow of blood in the specific area. Then once you felt it, sterilize the area with a 70% alcohol swab to remove any form of bacteria or particle, to sterilize the site of prick, to prevent infection. Then insert the needle of the syringe at an angle of 30 to 45 degrees depending upon the position of the vein or site. Anchor the vein once you target it and then insert the needle of the syringe into the skin to perform an intravenous incision. Then, after confirming the blood is inside the needle, slowly pull back the syringe to allow the blood to get inside the syringe. Once a required amount of blood is drawn, place a swab on the site of the needle and firmly apply pressure while releasing the syringe slowly out of the vein. Ask the patient to hold the swab after draw, except a tourniquet is applied before the draw and onto the arm above the site of the venipuncture and released after the blood is drawn into the syringe. The tourniquet allows the vein to become more dilated so it could be more prominent to see and draw blood. Once the blood is drawn out, act quick as the blood will start to clot. Insert the needle like this so it allows the blood to rush inside the tube and to be stored. Then gently invert the tube to mix the additive along the blood. In some cases, hemolysis can also occur during the time of draw. If the tourniquet is been held for too long or if excess pressure is applied from above of the syringe while the blood is being entered into the tube. Now the tube is ready to be labeled and transported to the lab. This was just a small demo. Now to actually draw blood, I did it off screen and I drew some blood from my older brother. These are two samples of his blood and let us perform several tests. So I loaded the yellow top ones into my centrifuge and centrifuged it for 5 minutes. After 5 minutes, it separated into two layers, where the upper layer contains plasma or serum, and the lower layer contains red blood cells. So using a micropipette, I took out some serum and transferred it into a small container. Now, this can be used for several tests, including hormone tests, routine chemistry detection of viruses like hepatitis B, C, and HIV. So, I took out a combi stick bottle to test it for several things. I took a dropper and took out some serum and placed one drop onto each and every block. The same was repeated for the other ones too. Then I let it sit for about 1 minute, and after 1 minute, it showed me some results. A trace amount of glucose presence of proteins, and the pH being alkaline. When I checked the other one, they also showed a 3 plus amount of protein and an alkaline pH. This is because blood itself contains several proteins like albumins and globulins, which makes it maxed out, while the pH remains slightly alkaline because blood itself is slightly basic between 7.3 and 7.4, while a trace amount of glucose is normal or nil in blood, making the results being normal. As for the other strip, it also shows a substantial amount of protein along with the pH being slightly alkaline and the rest remaining nil, meaning that it is just plain normal serum. Now, as for the purple color top tube, it contains an additive called EDTA, short for ethylene diamine tetraacetic acid, whose main function is to bind with calcium, making it much more effective for preventing the blood from clotting. Where the blood contains 13 clotting factors and calcium is one of the main clotting factors, which upon being occupied allows us to run several tests on it like CBC, HbA1c, blood group, and smear analysis. For now, I will figure out the blood group and perform smear analysis from this tube. As I said, blood itself contains antigens and antibodies, which help figure out a foreign pathogen. Our blood can be divided into four main types: A, B, AB, and O. Blood group A contains A antigens and anti-B antibodies. Blood group B contains B antigens and anti-A antibodies. Blood group O has no antigens, but it does have both anti-A and B antibodies, so you cannot let it mix with other blood groups, which makes it a universal red blood cell donor because it can donate blood to any blood type. Blood group AB contains both A and B antigens but does not contain any antibodies, which makes it a universal acceptor of blood because it does not contain any antibodies to trigger an immune alarm reaction. Another discovery was made by Carl Landsteiner and Alexander S. Winner through experiments with blood of rhesus monkeys in 1940. They predicted that our blood can also be divided by the presence of another antigen called the rhesus factor, derived from the rhesus monkey as rabbit serum was injected with monkey red blood cells, also agglutinated or clumped in human red blood cells, which helped understand unknown transfusion reactions and hemolytic diseases of the newborn. So, a person can be either Rh positive or Rh negative. Therefore, Rh positive can donate to their respective Rh positive and vice versa with their Rh negative blood types. This test is going to predict my brother's blood type. So I took a slide and placed 3 drops of blood onto the slide. Then I placed 1 drop of anti-sera onto each and every blood drop. Anti-A shows no agglutination as well as anti-V, which means it does not contain any A or B antigens. However, it does contains the Rhd protein, meaning the blood type is O positive. My blood type, however, is B positive, and this is because I get my blood type straight from my mother. My brother got this trait from my dad, who is also O positive. Now, however, this was all about blood grouping, and let us talk about smear analysis. Smear analysis is a laboratory test where a thin layer of fluid sample, usually blood, is spread across a glass slide, stained with dye, and examined under the microscope. Smear analysis can help us diagnose anemia, infections, blood cancers, and several diseases related with blood. To do this, I took out a glass slide and placed a drop of blood and smeared it to the end. Then I let it dry and fixed it with some methanol and let it dry. Then I prepared the staining solution by mixing 2 ml of water with 1 ml of blood stain, and I poured it onto the glass slide and stained it for about 10 to 15 minutes. After then, I washed it and let it dry and loaded it into the microscope. At 45x, we can see several cells and structures, but to properly see the RBC morphology, I switched it to the 100x. Here are normocytic and normochromic cells with a neutrophil and regular platelets. Here I can see some ovulocytes along with some fields containing elliptocytes. Well, overall, the smear remains normal. Just for a common observation, I took a sample and placed it onto the slide, and heated the remaining tube. This caused the blood to char and turn solid. I took another sample and placed it onto the position and covered it. Then I loaded both samples onto the microscope. This one, before heating, shows normal biconcave discs of red blood cells stacked upon each other, while the heated ones shows severe morphological distortion, specifically crenations. When red blood cells exposed over 47 to 50 C denatures and deforms into spiky and scalloped edges, known as inchinocyte or burr cells. A demonstration is that what happens if blood is exposed to high temperatures. Well, this was it. I hope that you learned a lot, and if you liked watching videos like these, consider subscribing and liking my channel, and I will hopefully see you in the next one. Thanks for watching.